Hello everyone, thank you for attending my presentation. My name is Jun Xia. I'm from University of Buffalo, in the Department of Biomedical Engineering. The title of my presentation is Photoacoustic Dual Scan Mammoscope. So this is a system we made in my lab, and I'm going to talk about the recent progress in the system development and also the patient imaging results. So before we talk into my current setup, I would like to give an overview of breast cancer. So commonly used breast cancer imaging techniques include X-ray mammogram, MRI, and ultrasound. So I think all of the audience are familiar with this. So quickly just summarize their limitations. So X-ray has low sensitivity to patients with dense breasts. MRI is very expensive, requires contrast injection, and ultrasound has a higher false positive rate. So that's why a new imaging technique is really needed, especially for patients with dense breast. Photoacoustic imaging is a relatively new technique when you compare with X-ray or MRI. So photoacoustic imaging is based on the photoacoustic concept or principle, where a short laser pulse is fired into the tissue, and then the energy will be absorbed. So once the energy is absorbed, it will generate heat, and the heat will create thermal expansion and generate acoustic waves. Because the acoustic wave is basically a mechanical wave, so any expansion will generate a mechanical wave. If we use a short laser pulse, then the generated wave will have a high frequency, which is the ultrasound wave. By using detectors placed around the tissue, we'll be able to detect where these waves are generated. So in other words, photoacoustic imaging allows ultrasound transducers to see the optical absorption inside the tissue. So instead of the mechanical contrast provided by ultrasound imaging, photoacoustic imaging sees the optical contrast. Common absorbers inside the human body include blood, lipid, melanin. And photoacoustic is a completely non-invasive procedure similar to ultrasound. And we use the endogenous contrast, meaning we don't need to inject any contrast agents into the human body. So there has been many photoacoustic researches going on so far. And in a recent review we did in the lab, we conclude there are a few photoacoustic features of breast cancer. So the total hemoglobin concentration is one feature because fast-growing tumor associated with a higher level of blood. And then oxygen saturation is another feature because tumor regions typically have hypoxia due to the fast growth of the tumor cells. And then vessel density is also a feature because tumor regions typically have higher number of vessels in order to feed the growth of the tumor. And peripheral vascular structures. So this is typically showing in large tumors. So when you cannot see the detailed microvessel in the tumor, you can still see the feeding vessels. So the peripheral vascular structure around a suspicious lesion is also a kind of important feature for photoacoustic detection of the cancer. So as I mentioned, there are several photoacoustic imaging systems has been developed. And so why we still need my system? So this is a quick review of some of the limitations of existing systems. Some systems have a limited imaging depth. That's because of the light attenuation. And some systems are bulky, require a patient bed, which takes lots of clinical space. So it's good for like big hospitals, but if we want to image patient in the clinic, so the smaller the device would be preferred. And some of the systems don't capture ultrasound data; they can only capture photoacoustic data. But we all know ultrasound is an established tool for breast imaging. So by doing photoacoustic, we don't necessarily mean we need to get rid of ultrasound. We can combine with ultrasound to provide more comprehensive analysis of the breast tissue. And some of the systems are subject to motion artifacts, and also the scanning time in some of the systems are also very large. For instance, one of the systems takes more than 10 minutes to image a single breast. So let's now introduce our DSM system. We call it as a dual scan photoacoustic mammoscope. So the reason we call it dual scan is because we will have two scanning probes, one from the top and one from the bottom. And we'll have the breast compressed in the same way of an X-ray membrane. But instead of using metal plates to compress breast, we use plastic membranes. So our compression is very gentle. And in all of our patient imaging, we never heard a patient complaining about the compression. So in a slightly compressed breast, we have two scanning heads scanning like through the breast to give a three-dimensional image of the vascular structures. And the whole setup is sitting on a lift table to fit patients of different heights. So the complete 3D scan takes roughly 1 minute. And then the lateral resolution, so lateral is along the transducer element direction, is roughly 1 millimeter, and elevation resolution is also roughly 1 millimeter. So now this is a closer look of the system. So here we have the transducer design, okay, which I'm going to show in the next slides. And you can see we shine the light into the tissue, and then we scan the transducer and the fiber bundle to give us three-dimensional view. The parameters, we use ND arc laser with 1064 nanometer output. The laser is a pulsed laser. As I mentioned before, pulsed laser give you the strongest photoacoustic generation. And we use two linear transducer arrays with 2.25 MHz central frequency, and the light illumination is well within the safety limit. And the scanning time is roughly 1 minute, and the scanning field of view is about 20 centimeter by 9 centimeter by 7 centimeter. The scanning head. The scanning head is one of the innovation of our design. So we have the transducer and the optical fiber. So the optical fiber illuminates light, gets reflected by a specially made dichroic mirror, and then goes through another mirror. So the first mirror reflects the light, and the second mirror is transparent to the light. The second mirror also acts as a reflector to collect the photoacoustic signal. By using this design, we make sure the light illumination and the acoustic detection are coplanar to each other to improve the signal generation efficiency. And this is a quick photo of the transducer. We use a 128 element transducer with pitch 0.67 millimeter, and the total field of view is roughly 8.6 centimeters. For ultrasound acquisition, we use a wide beam acquisition, so in which there are 14 scan lines at 3 stereo angles, and we find it to perform better than the plane wave imaging. So this is an image showing on a breast phantom where the wide beam acquisition code allows you to see the lesion very clearly. So this is a video of half of the. So you can see we can scan across the object, and here the object is the human finger. And after scanning, we can get a three-dimensional view of the vascular structures. So again, the contrast comes from blood, so we don't need to inject anything. And the contrast, once it's reconstructed, we use depth encoding, meaning different depths shows displayed in different colors. So here are some results of our breast imaging system. So after we image multiple patients and healthy volunteers, we reconstruct the data and project it in the same view of an X-ray membrane. So it's a CC view. So in this way, it makes it easier for radiologists to read the image. So you can see the image is depth encoded. So here, like the blue vessel means shallow vessel, a blue color means shallow vessel, and then the red color means the deep vessels. And we have the right breast and the left breast. and in the middle is supposed to be the chest wall because we combine the data in the same view of mammogram. So this is the data is included in our first paper about this system, which is published in IEEE Transitions on Biomedical Engineering. So in comparison to X-ray mammogram, and we found our system is not affected by the breast density. So we have imaged patients, and we have the density classification of these patients. So from these image results, you can see X-ray, as we all know, is affected by the density, but the photoacoustic imaging vascular structure is not affected. And we also compare with contrast enhancing MRI. As I mentioned, photoacoustic imaging does not need any contrast injection because the blood serves as an intrinsic contrast, but MRI requires. From the MRI, you can get the vascular structures, and after comparing the MRI with the photoacoustic results, we can find the same like branch of the vessels. So in this way, we can confirm that we show similar vessel structures as MRI. So actually, you can see from this picture, we actually show denser vessels than the MRI, but without the need of any contrast injection. And here is a comparison with clinical ultrasound. The clinical ultrasound, we all know, operates at a much higher frequency. And with our 2.25 megahertz ultrasound, the frequency is lower, but we can still identify like the suspicious lesion. So this is an example from the patient imaging results. We'll compare with the clinical report. The shape of the tumor looks very similar. And from the vessels, so here the color in yellow shows the vessels captured by photoacoustic. From the vessels, you can clearly see there are rich peripheral vessels, which acts as a feeding vessel to this tumor lesion. And this result actually agrees with another study conducted by other group, which confirms for a luminal A tumor, so you will be able to see the peripheral vessels, but the internal structures, like the internal photoacoustic intensity, is low compared with the surrounding tissue. We have also done a statistical analysis based on the patients we imaged. We have imaged 38 patients, so that was the date like of roughly like 1 year ago. Okay, so after we capture 38 patients, we do a statistical analysis. So the way we compare the data is we analyze the photoacoustic features of the tumor and the healthy breast of the same patient. So each patient, we image both breasts, and then we try to see if there are any difference between the two breasts. So we extract some of the parameters. One is called the vessel mean ratio, which quantifies the average vessel signal within the whole breast. The contrast ratio compares the contrast of the tumor or the contrast of the vessel versus the background. So in the tumor case, it's a vessel versus background. In the healthy case, it's also. And then we compare the two. And then the other is the standard deviation, which compares the variation of the vessel signal. And then the standard deviation of the background, which considers the variation of the background signal. And so we can have data pair for each patient and each subject because we can analyze both of the breasts. So based on the statistical analysis, we found out that the P vessel mean, so means the average vessel signal in the tumor is in the tumor bearing breast is higher. So that means the presence of larger vessels in the cancer's breast. The contrast ratio is also higher, and this agrees with the finding on the first one, which is the mean vessel density. And we also found the standard deviation in the background is higher, so that means there are strong variation in the tissue signal. So our hypothesis is maybe caused by the growth of the microvascular structures. And these conclusions was done in only 38 patients, but they agree with the published literature on the tumor angiogenesis regarding the breast cancer. So some of the conclusions are not very significant with a p-value larger than 0.5, but they're very, very close. So we think once we increase the patient number, we'll be able to get a more conclusive conclusion. So right now we're still continuing imaging the patients, and here I'm going to show some of the recent developments we made in the lab. So the first is we called the second generation DSM. And in this DSM system, we use a round trip scanning to, to, to get a larger field of view. So once we scan like from the chest wall uh, away from the chest wall, and then it will return to the chest wall. So during the round trip scanning, we'll shift the transducer to provide a larger field of view. So I'm going to play a video here. So you can see first away from chest wall, do a lateral shift, and then scan to the chest wall again. So you can see it's running in continuous motion. So in this case, we get a larger field of view, allowing us to cover the entire breast. So you can see first trip gives you half of the breast, second one gives you the other half. By combining both, you get a much larger field of view. So the second generation system was published last year in Frontiers in Oncology. So move to the next one. And in this second generation system, we'll also add ultrasound elastogram. And to do so, we use like a motorized compression stage. So let me review all the sides. So we use a motorized compression stage. So the compression stage is going to apply a 1% compression. And by capturing data before and after compression, similar to a conventional ultrasound elastogram, we're going to see which tissue is harder, which one is softer. So by doing this, we characterize both the photoacoustic contrast, right, and also the hardness of the tissue. So this is the image we captured on a breast spanton with a hard lesion in the center. Using this technique, we're able to characterize which part of the tissue is harder. And we're also using deep learning to further improve the resolution. So this deep learning result, this deep learning algorithm, we name it as DPE, was published in IEEE Transitions on Medical Imaging. So in this study, we use a very novel approach to improve the data in 3D, even though our training was done on 2D. So this allows us to reduce the amount of the training data, but still gives you a very good performance. So here it shows the deep learning improved the results. So the input is here. So this is the input from this is the original reconstruct data from our DSM system. After inputting into the DPE, you can see a much higher spatial resolution. You can see the vessels become much sharper. And also some of the artifacts and background noise can also be removed. And this is another input. You can see the vascular structure is improved greatly. So this technique allows us to get much finer vascular structures and also a cleaner background. So we recently also combined shear wave elastogram into the system. So the quasi-static elastogram mentioned previously requires a compression. So it can only be done on a single frame. So in this one, by using shear wave, we can continuously scan across the breast tissue. So in this case, we do photoacoustic, wide beam ultrasound, and also shear wave. So on the one trip, we do photoacoustic and wide beam ultrasound acquisition simultaneously, and then on the return trip, we do the shear wave. So in shear wave, we send a push pulse, and then we use track beams to track the propagation of the push pulse. And by analyzing the shear wave propagation, you can find out which lesion is harder, which one is softer, because the shear wave propagation speed will be different. So we validate the scaling mechanism in a phantom. We see the photoacoustic feature, which is a dark tube. We see the ultrasound feature, so which is an ultrasound-like lesion with a high echo reflection. And then on the other side, there is also a lesion which doesn't show up in ultrasound, but it has a harder inclusion. So it shows up in the shear weight. So by using this technique, we can see three different distinct features. And finally, I would like to thank our team members. So my students, I have three PhD students, Nikila, Hujun, and Emily. So one has already graduated. They contribute a lot to this study. And I would like to thank our collaborator, Roswell Park Comprehensive Cancer Center, which gives us access to the patient. And our study is funded by NIH, Susan Komen Foundation, and Winston Radiology is also a collaborator, which helped us at the beginning of the project. Thank you for your attention, and I'm open for questions.